All right, digestive system, physiology. You're going to find that a lot of the stuff that we covered in anatomy is also going to this. It'll come up again here, so that's kind of good. And again, you know, if somebody else were teaching this, would they include any anatomy in it? You know, I come from an undergraduate background where I taught anatomy and physiology for like 100 years. That's how my mind thinks. And when I'm going through the digestive system, I'm not going to talk about, hey, secretion. Here's all the things that are secreting. And then, oh, hey, here's absorption. I'm going to go through the digestive tract from your in-hole to your out-hole. Because that's what makes sense, right? So that's how we're going to talk about it. So here's an overview of the digestive system, showing you all the most of the relevant parts, how long each one is. I'm not going to ask you questions like how long it is. That's like a stupid undergrad trivia question that you get for extra credit. But so we're going to follow that too, and I'm going to impress upon you right away that when you're inside the tube, you're still not actually inside your body, right? Because you can snake a string through, swallow it, and come out the other end and go wee, wee, wee. And it still connects to the outside of your body, right? Don't try that. But anybody who has ever had cats, they do that sometimes, don't they? Like I have done that. I've had a cat swallow something, it's out this end, but they're still like puking it up this end. Let me just pull it out. This cat's a little anything. All right, so mainly the focus is going to be physiology though. So when we're talking about the digestive system, physiologically speaking, we're talking about how food is broken down and how we process it, absorb it, and spit out whatever's left over. Okay, so also called how to make food. So we have two types: mechanical digestion, chemical digestion. Mechanical digestion, of course, is a good example here. You take a bunch of food in a bag and smash it with a hammer. That's the same thing your teeth does, right inside your mouth. You don't alter the chemical properties, so that's just breaking it down into smaller chunks. And then chemical digestion is taking those smaller chunks and breaking it down into chemical. This is the same problem we're having the other day, wasn't it? Small enough chemicals to be absorbed. All right, so you want to take like nice soft chocolate chip and break it down into individual monosaccharides to be absorbed. Okay, so again, the overview. We're going to start with the mouth, talk about the digestive properties that go in there, the food tube, stomach, small intestine, large intestine, and out to okay. We're only going to get through about half of this today, so I don't remember where that takes us, but we're going to try to follow that as much as possible. What I gave you as a handout are most of the hormones that you'll find secreted by the digestive system. Now, when we're talking about, sorry, I'm just doing your thing. When we're talking about hormones, where are they secreted? Where are hormones secreted? Are they secreted into the digestive tract? Into the blood. So all of these hormones are things that are going to go into the blood and affect the digestive tract. So don't go thinking like, oh yeah, our stomach makes gastrin and it just secreted to the stomach and does stuff. No, it's got to go through the whole circulatory system and come back to the stomach. So one important thing to keep in mind. The reason why I printed it out for you is out on canvas for you as well. In fact, there's down at the bottom, there's a little dash and a 6. That's a typo. That 6 doesn't mean anything. And I got rid of it on your I got rid of it on your on campus, so if you print it out for yourself or look at it, you won't see it anymore. So that's how we'll go through it. In talking about the alimentary canal, right, which is another name for the food tube. Say that's a tube. And here's your lumen, all right? And from the outside to the inside, there's 4 layers, all right? Actually, since it's that way, from the inside to the outside, there's 4 layers, all right? First of all, coming in contact with anything you swallow, so this would be the lumen, but the layer that surrounds that lumen is called the mucosal layer, all right? And in the alimentary canal, the mucosal layer is going to be specialized in each area of the digestive system for doing specific things. So for example, in the stomach, the mucosal layer is going to be more designed to do what, do you think? Pardon? Protect, but in terms of digestion, though.
Be acidic.
Be acidic, pardon?
I just have smaller.
Secretion. It's going to secrete things, right? But then when we get down into the small intestine, that's mucosal layer is more geared towards absorption, right? And then we get to the large intestine, and that's geared towards like kind of a dual layer secretion and absorption. But of different things. So that's going to change all along the digestive tract as you go along. And esophagus too, I guess that's part of the food tube, and that's again secretion because not much actual digestion goes on in there. All right, just outside of that, we have the submucosa. And it's called submucosa because it's underneath the mucosa, even though we're going outside. Does that make sense? So submucosa, and that is where a lot of your some nerves, blood vessels, things like that are going to be located. That will help supply the tools, if you will, necessary for the mucosa to do its job. All right, the third layer outside of that would be the muscular layer, and recognize that you have 2 layers of muscle. We'll do this brown layer. Has nothing to do with the actual what's in the digestive tract. And that's going to be your circular layer. And then we have your longitudinal layer. I really did not draw this to scale. So, all right, I gotta tell you guys a story. I was walking down the hall the other day with this, and one of my students from last year saw the drawings on there, and he's like, "Did you draw?" No, wait, you definitely didn't draw those. It's like, what? Hey. So, my artwork. So, a circular muscular layer, then the longitudinal muscular layer. So the circular muscular layer would take the tube and squeeze down on it, right, at a specific location. Then the muscular or the longitudinal layer would move things down in the direction, one direction. You're going to find in the stomach, though, that there is an additional layer, just because of the stomach's function. And then finally, on the outside, green. Actually, I'll do it purple. It's purple already. The outside, you'll have the serosal layer, and that's going to be connective tissue, part of visceral peritoneum that holds everything together. All right, we've already talked about this, mucosa. This is just words for what I've already talked about. This is showing you in the stomach what a micrograph of the stomach would look like, and you notice that there's not as many, it's not as textured as the small intestine because all this stuff is glandular and it secretes stuff into the lumen of the stomach, whereas this would be the small intestine. Here you see the villi, which increase the surface area for absorption. So very different appearances of the mucosa. right? Submucosa, what it is, what's in it, we've already talked about that. Muscular layer. See, I get ahead of myself. So all this is stuff that we've already mentioned. All right, so now let's get into some of the physiology. If you've already gone ahead and looked at the objectives for today, the instructional objectives, the revised ones that are in Canvas, I changed them.
Again?
Huh?
Again?
Yes. So what I did is I went in and I just uncrossed one thing. And that one thing is related to this stuff right here. So it's not a big change. In your digestive tract, you have muscles, right? And those muscles have to contract. But in order to contract, they need a signal, just like the heart does. So there are rhythmic signals caused by certain cells within the smooth muscle of the alimentary canal that produce what we call slow waves. All right, and these slow waves are at a fixed frequency. So if we draw this on a graph, we have going like this, and let's say this is threshold right here. We constantly have these waves going like this all the time within our digestive tract, just under threshold. So what that means is if it's under threshold, they're not actually stimulating an action potential. So they're not actually stimulating muscles to contract. All right. These slow wave signals come from these cells called the interstitial cells of K-Hall or K-Gel. I've heard it pronounced both ways. ICC cells, we'll call them ICC cells. Those are the pacemaker cells within the digestive tract. So we talked about pacemaker cells within the heart, right? Where do those come from? SA no. All right, these are the pacemaker cells of the digestive tract. So there are some within the interstitial cells, and there are some embedded within the actual muscle tissue as well. And what these do is they constantly do these slow wave electrical signals repeatedly. And the reason, well, let me see this. The reason for this is what caused the rhythmic contractions in the heart? What was responsible for the automatic fires in the heart? Leaky sodium channels. All right. Here we have leaky calcium channels. All right, leaky calcium channels. All right, so if we have calcium, here's a cell. Where's the highest concentration of calcium? Inside the cell or outside the cell? Outside. So if we have leaky calcium channels, where's calcium going to go? Inside. Okay. So leaky calcium channels. And then when the cells get just below threshold, potassium channels open, and potassium goes out, which is why the wave goes back down. All right? So that's constantly going over and over and over again, happening independent of nervous system stimulation, all right, fired by these. All right, the other thing to take note of is that between these ICC cells and the smooth muscle cells, there are also gap junctions. What do those gap junctions allow? Contraction at the same time. Movement of ions between cells without any interference. All right, so you have that within the smooth muscle cells and within these ICC cells as well. So what happens is you have these slow waves always going, and then something happens. The nervous system intervenes. Which branch of the nervous system intervenes to make these waves go above threshold? Parasympathetic. Good. Parasympathetic, rest and digest. So they're going to cause either those or hormonal stimuli, right? So certain hormones stimulating the getting old. smooth muscle cells will cause it to go just above threshold. And then when it goes above threshold, you fire action potentials. And then it comes back down. Do that. Okay? We call those spike potentials. Right? The stronger the stimulus, in other words, the higher this goes, the higher the amplitude goes of these slow waves, the more frequent those spike potentials are going to get, and the stronger the contraction. Right? So the more stimulus, more frequent spikes, stronger contraction in the smooth muscle. All right? So this is a picture of what we were talking about. These are the slow waves right here. Then you get a nervous system intervention. It makes it go a little bit higher. You get a few spikes. Then you get some more stimulation, even higher spike. Notice that there's more spikes, higher frequency. So this would give no contractions, weak contractions, strong contractions, for example. Okay? All right, and again, this is all moderated by calcium, not sodium. Okay, so a little bit different than what we've learned with the cardiovascular system. So, what kind of things? We already talked about acetylcholine, parasympathetics, anything that acetylcholine is part of the nervous system that would trigger them. Parasympathetics, of course, the parasympathetic nervous system. Also, stretch of the smooth muscle can increase the amplitude of these slow waves. All right, all to produce these contractions. All right, and then this says the same thing that the previous slide says. So why I have it here, I don't know. Anyway, if we have, we also have the sympathetic nervous system that goes into the gut as well. That has the opposite effect, though, right? If we have the sympathetic nervous system mediated by norepinephrine, what did I say about acetylcholine? I don't remember. Acetylcholine is the neurotransmitter for parasympathetic, right? So, you guys remember from the first couple of weeks? Wait, just go back a second, sorry. When we have the sympathetic nervous system, we have a short peak preganglionic and a long postganglionic, and then parasympathetic, we have a long preganglionic and a short postganglionic. This is going to, I'm drawing this also now. I want to cover this now because we're going to talk about it a little bit too. Here, our neurotransmitter is acetylcholine. Same thing here. Here is where we have norepinephrine and acetylcholine. And the difference is the receptors on the target cells. Okay, so acetylcholine would stimulate, be the stimulus from the parasympathetic nervous system. So if we have norepinephrine, that's going to cause hyperpolarization. So what you will see with these waves is they will get even smaller. Right? So they're harder to get up the threshold. And that's what the sympathetic nervous system does, is it decreases these slow waves. So what we might mean by sympathetics and parasympathetics are drugs. Like we're providers, right? We're going to be giving drugs. If we give sympathetic stimulating drugs, they will have a hyperpolarization effect. If we give parasympathetic stimulating drugs, that will have a depolarizing effect. Right? Does that make sense? I mean, I know a lot to go through, but let's review. Somebody asked me about testing as far as the first 2 weeks of material, which is what some of this is, right? I'm drawing on a board. You're done testing for that material. right? I'm not going to test you over again. You've had 30 questions worth of week 1 and week 2 material, which is half of an exam, if your exams are 60 questions. So, but stuff like that is going to come up again. So I'll bring it up again here, and you might see it on test questions related to what we're talking about, but I won't directly test you over that material. Does that make sense? Okay. All right. Because smooth muscle cells have these excuse me, gap junctions in them, when we stimulate one cell, and say this would be an ICC cell, it shares a gap junction with a smooth muscle cell, which then shares gap junctions with other smooth muscle cells. They all contract as a single unit, right? Because of that calcium flow through into smooth muscle cells. And we call that a syncytium. Even in the heart, that's called a syncytium, where everything's contracting as a single unit. All right, so that's what the electrical activity looks like. But when we have contractions going on, what do those contractions look like? So we have two types of movements basically in the alimentary canal. One is a propulsive movement, and by propulsive we mean moving along in one direction. We refer to that as peristalsis. Right? After you had a good lunch, you're like, oh, there goes peristalsis. All right, so what happens with peristalsis is actually what stimulates peristalsis. One, distension. If you're swallowing something and your tube stretches, that tells, remember, stretching was one of the impulses for getting those slow waves up to spike potential. So when your tube stretches, the smooth muscle cells are going to go, oh, hey, we need to contract, right? So spike potential start, and you get this slow wave contraction down the tube. Irritation of the gut epithelium, right? When you go to BW3s, any hot wings, and all of a sudden you feel like, oh, I gotta go to the bathroom, I'm sorry, right? We've all experienced it. That would be irritation of the epithelial cells of the gut, right? So that's going to start moving things along. Peristaltic rush. As well as the parasympathetic nervous system, right? When it's time, like you swallow food, parasympathetic nervous system's in effect, and it's going to move things along for the next couple hours. So this is what it looks like. What happens during peristalsis is you swallow something that stretches. Ahead of the contraction, you have what's called receptive relaxation. So the area in front of whatever is moving in the tube has to relax in order for the area behind it to push it along, right? Because what would happen if you contracted in front of and behind? It's stuck. It's not going to go anywhere. So you need receptive relaxation ahead of where the direction of the movement is going to go. And generally speaking, peristalsis only occurs in one direction. Oh, so I don't know why I don't have this. The other type of movement we have, so propulsive, would be mixing movements. All right, and this is what a mixing movement would look like. So you have, assume this was all a green mush. We have contractions before it, contractions, behind it, before it, behind it, before it. They segment, and then they start mixing. They segment and mix in different areas, and that kind of chops up your bolus of food. All right, and that's what we call a mixing movement. And everything mixes together in a nice homogeneous mush. Which makes me wonder. My son, you guys get all kinds of stories. They did, you guys remember this from undergrad or maybe high school, they did owl pellets. Dissected owl pellets, which it was fun. But my son was like, man, these guys that I was with, they didn't let me do it. They did all of it themselves, and I just had to watch. So he was kind of bummed out about that. So I'm like, science, Dad. to the rescue. So you go on Amazon, they'll sell they'll sell owl pellets. So I bought 6 owl pellets, like $25. I was like, that's pretty expensive. Whatever. And so you get these, and then you open them, and who has cats? They're basically just a hairball, right? But they still have skulls in there and bones and whatever. And so that makes me wonder, how much was all of that mixed up? Like if you still have intact skulls. That's just weird. And then all of that to come back up.
Did you imagine a skull like going up your throat to come out?
Yeah, no.
So gross.
But it's interesting because when you open these pellets, so when they come out, they're wrapped in mucus too. But like it seems like the fur is all around the outside and everything is on the inside. And I'm like, how do they do that? That's like amazing. Anyway. So how do we get the nervous system involved in the control over these occurrences within the GI tract? So we have 2 types of neural control. One is intrinsic. You guys have probably all heard at some point in your lives that we have the digestive system has its own brain, right? That's what we call the enteric nervous system, and that is entirely within the tube, entirely contained within the tube. So it can function independently of the parasympathetic nervous system. And there's 2 plexuses that you should be familiar with. I'm not going to use the names. I use, their functional name. So the myenteric plexus, anytime you see the prefix my, what does that usually refer to? Muscle. And then the other is the submucosal plexus. And what does that stand for? Submucosal. All right. So that tells you exactly where they're located. One between the circular muscle and the longitudinal muscle, and the other one is entirely within the submucosum. All right. If you had to guess, what's the function of the myenteric plexus? Movement, contraction of that smooth muscle. What's the function of the submucosal plexus? Secretion. Right? So I'm totally okay if you guys call the mucus food lube. Food lube. Apply some food lube. What? Oh, yeah. Then you have extrinsic control. I mean, those words, intrinsic, entirely within the digestive system, extrinsic, entirely outside of the digestive system. And that's the autonomic. So you have your parasympathetic, stimulates activity, parasympathetic, inhibits activity to the actions of acetylcholine and norepinephrine. What's another name for norepinephrine? Just for noradrenaline, right? Adrenaline, epinephrine, noradrenaline, norepinephrine, same things. Which always, I'm going to get on this about for a second. So why do we call it norepinephrine, but it binds to adrenergic receptors? Why don't we just call it adrenaline and just make it easy? Right? You guys ever wonder that? I mean, they have committees to discuss this stuff. They do. They literally do. World committees to discuss this stuff, and you would think they would have gotten their shit together by now. All right. So, there's a good example. So we have, this is where they're located. So here's your submucosal plexus, within the submucosa, and then your myenteric plexus between the longitudinal and circular layers. All right, so this is just showing you where they're located. Okay, and this is kind of showing you a picture of, so if you imagine your longitudinal layer of muscle would be right here, and if I had a board here, I'd draw, your circular muscle layer would be right here, and here's your submucosa right here. So it's showing you how, one, they're interconnected to each other within a plexus and connected to between each other, between plexuses, and here's where your sympathetic and parasympathetic are going to innervate, right? So these can all happen independently, but the sympathetic and parasympathetic nervous system do directly go to them to stimulate them to go above and beyond what they're normally doing, okay? Also recognize that you have sensory neurons coming from the mucosa that will go to the central nervous system and say, hey, we need you to intervene, and that's when they'll start getting involved. All right, we're going to see examples of this once we get into the actual specific functions of parts of the alimentary canal. Any questions so far? No? All right. So where does the innervation come from? Mostly your vagus nerve. So the vagus nerve, which is cranial nerve, anybody remember? Or no? 10, right? Vagus nerve, cranial nerve 10. That's going to be most of your GI tract. You're going to see some cases where that's not true. So keep that in mind. Sacral division, pelvic splanchnic nerves, distal colon to the anus. Right, so the distal colon to the anus is not controlled by the vagus nerve. That comes from something else. We talked about the specific neurons from the parasympathetic. We drew them out right there. This is exactly what this is saying. So here relates to that. And parasympathetic excites. Okay, again, rest and digest. And then a picture showed you the afferent sensory fibers. All right, sympathetic innervation. They come from thoracic and lumbar vertebrae. Okay, and that's kind of covered in your anatomy as well when we talk about the sympathetic nervous system in that area. We talked about how they're set up in terms of the neurons and generally decreases activity and of the enteric nervous system, and also can directly inhibit smooth muscle. SM stands for smooth muscle. Because if we look back to this, we have a sympathetic right here, directly to smooth muscle. That's what this would be right here. Okay, we've talked about this. We've already gone over this. This is that picture right here. Let me add one thing to this. So, sympathetic neurons, the preganglionic releases acetylcholine. Do you guys remember what kind of receptors on the cell body of the postganglionic neuron? Pardon? Cholinergic, but specifically, what kind? Which is it? Not muscarinic. Have to be nicotinic. All right. Down here, it's going to be the same thing. Nicotinic, cholinergic. The postganglionic releases acetylcholine, but the effectors have muscarinic this time, parasympathetic nervous system, and norepinephrine has adrenergic receptors. Okay, so that's how they work. Within the enteric nervous system, there are all kinds of other neurotransmitters and receptors for them. And these are just a short list of what they are. All right, we're not going to get into it. I've gone over it. I did my job. Does anybody know what VIP and NO are? Vasoactive intestinal peptide.
Is the NO nitric oxide?
And the NO is nitric oxide. Vasoactive intestinal peptide. And if you remember, this tells you what it does. It acts on blood vessels. Nitric oxide also acts on blood vessels, right? So it's known as a vasodilator. All right, certain reflexes that occur in the the digestive tract. One, the vagal reflex. All right, I always want to say vasovagal. That would be something completely different. It's a vagal. All right, so the way that works is you have something going on in the stomach or duodenum. Actually, a good example of the vagal reflex, just get this before going ahead of it, is the enterogastric reflex. The enterogastric reflex, and we'll go over that later. So something happens in the stomach or duodenum that sends afferent fibers to the brainstem. The brainstem then sends efferent fibers back to the stomach or duodenum to do something specific. Okay? So that will involve both motor and secretory activity. Defecation reflexes. All right? Something happens in the colon or rectum. What do you think is happening there? Pardon?
It relaxes?
Well, you have to have a stimulus to relax. So what is going to cause a stimulus to come down and cause it to relax? What's the poop doing to the tube? Stretching, right? So stretching of the colon or rectum sends afferent fibers to the spinal cord. Spinal cord sends efferent fibers back to the colon or rectum, not to relax though, but to push. And you get defecation, right? And also pain reflexes that generally inhibit the GI tract. If your patient is in pain, they're not going to poop. That's one of the big pushes for why we are big about pain control, is because we want our patients to go to the bathroom. If they don't go to the bathroom, that causes all kinds of other health problems. One of the problems with that is opiates. Do we know what they do? They slow down your digestive tract, so they're not going to poop anyway. So let's get them off the opiates. And I'm out of a job with my nursing because most of my patients are opiate patients, but we have a much better outcome overall. That's another soapbox we won't get onto. All right, you've seen this picture before with cardiac pain. Remember, because the heart and the upper limbs develop in the same place embryonically. When you feel pain in your heart, you're going to feel it down your arm. So these are spots where you feel pain with certain organs and where you might feel it away from that organ. Okay, so referred pain. I'm not going to go into any real detail about that, but I don't think this is completely accurate for the digestive system either. But one thing I do want to point out, notice that the esophagus right here. So if you have heartburn, you also might feel that in your left arm, which is why heartburn is often mistaken for heart attacks, right? Or angina pain, because they have a large area of overlap. All right, this is your list of GI hormones, which I gave you a piece of paper about. So basically I took this list and put it on in Microsoft Word, added 2 or 3 more, and that's what your handout is, okay? So I'm not going to go over this because we're going to cover this as we go along. But we should actually cover every single one of those at some point throughout the next, I don't know what slide we're on, the next 60 to 90 slides.
What is the green and red? What does that mean?
I put it there for a reason last year, and I can't remember for the life of me why I put it there. I would say what it probably means, just looking at it, is those are the functions you're responsible for, right? Red means inhibit. Green means stimulate. Blue means it still stimulates, but you're not responsible for it. Does that make sense?
Yeah.
Thank you.
Yep. All right, and then these are two other ones, somatostatin and histamine. But I want you to recognize, and you can even make a note on your little sheet I gave you, that these are strictly hormones, which by definition means they go into the blood and then come back and have an effect. These are paracrines, and what do we mean by paracrine, generally speaking?
And they release like effects.
Local effects, right? They're still secreted, but the effects are localized to the area, all right? So they don't necessarily have to go in the blood. All right, and I guess I did put VIP in here as well. So these are just a couple more, and I think I included those on, and yep. So all of this stuff, this slide, the previous slide, and the slide before that, those are all within that sheet. And we will cover them, but those are summaries you can go back to. All right, splanchnic circulation. And my wife got really tired of me because there were some arguments in one of your anatomy labs how to pronounce splanchnic. I'm like, all right, pronunciation is splanchnic. Let's resolve this. And I went to 4 or 5 different sites, and they all pronounced it the same way, which was splanchnic. I think somebody was saying splanchnic or whatever. Anyway, that's what it's pronounced. It just means enteric, guts. Splanchnic means your guts. So what splanchnic circulation entails is your GI tract, your spleen, which we include in the GI tract because there's no real else to put. Pancreas and liver. All right, so your GI tract plus the accessory organs. The arteries that go to these organs are 25 to 30% of your cardiac output. So a quarter of your cardiac output goes to your digestive organs. It's quite a bit. All right. This is what's feeding them. You guys should be very familiar with celiac artery, superior mesenteric, inferior mesenteric, right? I gotta stop saying that like I just told you that. Shut up. All right, and specifically what they're going to. Remember, celiac artery, stomach, duodenum, spleen, things like that, and upper, superior mesenteric, midgut, inferior mesenteric, hindgut. All right, so we're by foregut, midgut, and hindgut, we're talking about the direction of food flow. All right, venous drainage. I really want you guys to know this because this is kind of a tough concept, and we're going to come back to it a couple times throughout this, but we're just going to introduce it here. All these arteries go to these organs. They supply them with oxygen and nutrients and stuff like that. Then they go to venules, veins, and all the venous drainage from all of these organs go through a hepatic portal vein. All right? So this hepatic portal vein does not go directly back to the heart. Where does it go? To the liver. Because the liver has to process everything that was absorbed from the digestive tract before it spits back out into the circulation. All right, so this hepatic portal vein accomplishes that. Right, you've got this whole complex network that the blood goes through in order to achieve this, and we'll talk about that later on. We have pictures and everything. It's really cool. Okay, there are cells in there that remove bacteria, so you have immune function. And then of all the nutrients that go through the liver, about half of them are stored there, right? And why are they stored there? Think about glycogen, right? That's where a lot of your sugars are stored until you get the release to break down glycogen to increase blood glucose, insulin glucagon pathways, so that if you need them later, you can use them. Oh, here's a picture. I forgot this was one. So this is showing you spleen. Your stomach would be right here, right? Intestines all feed, all the artery or veins feed into the liver, goes through this system, and these are the hepatic cells that are absorbing all of that stuff and spitting stuff back out in it. They all converge and go into the hepatic vein, and it's the hepatic vein that goes to the vena cava and back to circulation. So you have the hepatic portal vein from organs to liver, and the hepatic vein from liver to circulation. Any questions about that? Some people screw that up. I haven't had enough cohorts to say that you guys screwed up specifically, but if you have any questions about that, please think about them and ask. I'm just seeing where we are. 25? We're about on track. So if you guys haven't noticed, like some of my slides have numbers, some don't. I pulled from different sources, and to get them to number correctly is such a pain in the ass, like I gave up. And it takes a long time to do that. All right, control of gut blood flow, because we want, remind me, what percent of cardiac output goes to the gut? 25%. All right, but there are times where we need to increase that or decrease that. All right, so how do we regulate gut blood flow? All right, it's going to be proportional to local activity. So after we've had a meal, we're going to want more blood flow, right? In fact, blood flow can increase 2 to 3-fold. So we go from 25% to 75% of our cardiac output for short periods of time, which is why you eat a meal and then you try to go out for a run and you're like, God damn, that was a bad choice. All right, but it's only going to be for a short period of time. All right. So after you eat a meal, what specifically regulates that blood flow? So we have vasodilator hormones, gastrin being one of them, secretin being another. So we're going to see that gastrin, secretin, and CCK have major functions, but they also have minor functions such as vasodilation. All right, kinins, anytime you see kinin, that's going to be a vasodilator, and low oxygen is also going to be a stimulus for more blood to go there, and that's true for throughout the body, not just for the gut. All right, so that's hormonal related or physiological related. Nervous system related, increased PNS and increased parasympathetic is going to increase gut activity, which then increases blood flow. Sympathetic nervous system decreases blood flow. So you just eat a meal and you're camping and a bear smells it and then comes into your camping site and now you're stressed out. Sympathetic system overrides parasympathetic nervous system, right? So that would directly decrease blood flow, get it going to your muscles so you can escape the bear. All right? So this term down here, autoregulatory escape, means that if you have that exact situation where you have blood flow going to the gut and all of a sudden that gets shunted by the sympathetic nervous system activity, the parasympathetic nervous system can auto-regulate and get back to that high blood flow, okay, eventually. So it auto-regulates itself. Exercise and shock can also stimulate SNS activity. All right. So this is one of the villi, let's say in your small intestine, and this is what blood flow would look like. You have arteries going in, you have all of these capillary networks, but then you have direct shunts from artery to vein. All right. So when it says here under normal conditions that 80% of our oxygen is shunted from artery to vein, This is what we mean. It's going directly from the artery to the vein and coming back out. So under normal conditions, this area is only getting 20% of the oxygen that's available. Right? And that's all it needs because you don't have that many cells there. So it's only going to need 20% because this is like in contact with your gut on the outside. So the rest comes back. But if you happen to have a situation where you're not getting as much blood flow in general out to your body, including to the your gut, well then 20% of 100% is normal. You can live with that, but 20% of 50% can put you in danger, right? So that's going to cause these microvilli, if they're not getting the blood flow, to die. If they die, they atrophy. If they atrophy, what happens to your ability to absorb nutrients? Tanks, right? So this is why elderly people who have cardiac problems end up getting digestive problems because that blood flow is decreased to the point where their villi start to die off. The absorptive capacity decreases, and you got to watch what you feed them. That looks like a good stopping point. Why don't we take a break here, and we'll continue on.
That changes the song.
What country's not allowed?
Just scream it down the hallway. We're gonna think the nuts coming after us.
So I do recognize that not everybody likes my music, so I keep it to myself. But whatever. All right. Open lab, not class. Just like my wife, like feel good Christian type music. I love my wife, but somehow we get along.
Are you still recording?
I am. Oh, she knows. I said, turn that shit off. All right. Now let's get to the actual taking stuff in and pooping it out. All right. This is where it gets taken in, just for the record. And actually, it's funny, when I used to teach pathophysiology, we talk about when you have a blockage, and sometimes fecal matter comes out the other way, like that's really gross. And like you say, that student's like, ugh, that happened? That can happen. Gross. But, you know, the plus side is when they say something tastes like shit, they really know what they're talking about. So starting in the mouth, our cheeks, of course, our lips. Keep in mind that lips also have sensory receptors. That's why, you put that, a baby, you put that spoon and you touch their mouth there. That's a reflex. And then the tongue, of course, the tongue responsible for taste, but it's also responsible for moving things around. Next time you eat something, pay attention to actually what your tongue is doing. You'll see how much it actually moves and which directions it moves and what the food's actually doing when it does that. And then, of course, at the back of the mouth, we have tonsils. We have lymphatic tissue. And the goal of that is you're taking stuff into your mouth that's from the outside. Don't ever look at what you're eating under a microscope. Let's just put it that way, right? Because you'll be like, I'm never eating again. Like, just give me an IV solution of something sterile, and I'm good. So you have these lymphatic tissues that's going to monitor everything that's going down in your throat so that you can avoid infections. right? But then again, everything that goes down to the stomach is going to do what? Die anyway, hydrochloric acid. All right, so in the mouth, we have saliva. And saliva's job is to moisten and solubilize food, right? So this is what we refer to as food loop. All right. Aside from doing that, it also contains a lot of bicarbonate. Bicarbonate is a base. Base is buffer acids because what we want to take in our mouth is we don't want to be too acidic until it gets down to our stomach. So to protect, we have bicarbonate ions that buffer acids. Because also as we're breaking down certain things in our mouth, like sugars, they can produce acidic byproducts. We need that buffer in there to help minimize that, the effects of that. Saliva also has a hygienic effect. It has enzymes in it, such as lysozyme, the same lysozyme that's found in your tears that breaks down proteins from bacteria. Thiocyanate antibodies that help keep your mouth clean. Although, what do they say about your mouth? It's one of the dirtiest. No, I'll keep that story to myself. All right. To make saliva, we have two types of secretory cells in salivary glands. All right? I'm not saying there's two types of salivary glands. I'm saying there's two types of cells in each salivary gland. We have serous cells that are going to secrete serous fluid, and serous fluid is watery. All right? So that's what moistens food, solubilizes things. Things are soluble in water. All right? Those serous cells also secrete amylase, right? And amylase breaks down carbohydrates. So carbohydrate digestion begins in your mouth. That's why when you eat like a cracker or noodle and you leave it in your mouth long enough, it starts to taste sweet because amylase is breaking down those carbohydrates. We also have mucus cells. All right, mucus cells are secrete mucus. Mucus is thick. It's full of polysaccharides. That's what makes it have that thick consistency. And that binds things together and lubricates it so it's easy to shove down your throat. All right, so it makes it slide down easily. All right, what are the salivary glands that have these cells? Well, you have your parotid glands right here, so they're just anterior to your ear. All right, and notice that the ducts are going into the mouth here. There are specific names for that duct, which we will talk about in anatomy. That's not the purpose of doing that here. Submandibular glands underneath the mandible. They have ducts that go into the mouth, and then sublingual glands under the tongue. All right, most of your salivary secretions come from the parotids and the submandibular glands. All right, and they're mostly like a watery, mucousy consistency. The sublingual glands, strictly thick mucus fluid, all right, to help bind and lubricate things. And that's related to the digestive function. You want a lot of watery fluid with enzymes so you can digest carbohydrates. All right, innervation, sympathetic and parasympathetic, and it's really weird that your salivary glands are the weird part of your digestive system in terms of innervation. Sympathetic impulses can stimulate a small amount of viscous secretion, right? So sympathetic impulses can cause your salivary glands to start secreting viscous substance. What do you think the purpose of that is? Why would you want a viscous substance secreted when you're not eating things? So you don't dry it out, right? Prevents dry out. Then parasympathetic fibers, they secrete a large volume of watery saliva, all right, which contains the digestive enzymes, because that's when you're going to be starting to eat things, all right? So, how does that work? All of these things right here are things that stimulate salivary glands. These are things that inhibit salivary glands. And I thought it was weird. This isn't my slide. I stole it from somebody. When I first saw this, rough objects, and I did the experiment. take, like, go home and take, and this is just an example, you guys have to home probably, maybe you do, it's weird, I'm not. Like a jack, like, you know, ball and jacks. Take like a jack and put it in your mouth and see how fast your mouth dries up. Yeah, so rough objects will inhibit saliva, which, you ever seen Willy Wonka in the Chocolate Factory? And those, like, everlasting gobstoppers at the end that were all rough? Like, really? They last long because you don't have the saliva to... These are what I think of when I watch movies. All right? So, these are going to stimulate the medulla because where does your parasympathetic nervous system come from? I'm sorry, vagus nerve come from? The medulla, right? The medulla. So that also tells us that regulatory controls for digestive activities, including reflexes, also happen in the medulla. All right? That's going to activate or inhibit parasympathetics that go to the salivary glands and secrete substances, or go through that pathway and inhibit the secretion of these substances. Right? And then sympathetics act through, of course, a different way. The thoracic vertebrae going to specific superior ganglion cells, norepinephrine, salivary gland, stimulate thick secretions. All right, so this is just showing you the pathway and what stimulates. Somebody have a question? All right, and this is kind of a map of what that looks like. Anatomically speaking, so we have our tongue right here. Notice that that's going up here, through here. So these are all afferents going to the central nervous system. So if we go back, let me see. That would be here, right? This green or this red going to the medulla. And the medulla is going to come out and go to the salivary glands to cause secretion. But what do you notice on here that's different than what we said originally about stimulation of the digestive tract? Where does most of the stimulation of most of the digestive tract come from? What nerve? Vagus. Facial nerve. What does it say there? Glossopharyngeal nerve. All right. So salivary glands, not the vagus. Those would be the glossopharyngeal and the facial nerves. So it's a little bit different. But if you look, and I put this picture in just to show you, facial nerve and glossopharyngeal is still within the medulla oblongata. So still going to that area. All right, pharynx. We covered this, right, when we did the respiratory system, so I'm not going to cover that. As far as the digestive goes, we don't care about the nasopharynx, though, right, because we're not sticking food up our nose. So food is just going to go from the oral pharynx down into the laryngeal pharynx and down to the esophagus. All right, so we have saliva. We've chewed up our food. When we chew up our food with the saliva, taking the enzymes away, what kind of digestion is that? Mechanical, right? So we've covered mechanical digestion. Most of it occurs in the mouth. Now we want to get that bolus of food. At this point, it's called a bolus because we've broken it down, we've lubricated it. Now it's a ball of food. It's called a bolus. All right. And we want to get it down to the rest of the digestive tract. So we swallow. And swallowing is the other name for it's deglutition. So when you say deglutinate, you're swallowing. Happens in 3 phases. One is the voluntary phase. Our voluntary phase means you're purposefully doing this. All right? So we chew it. That's conscious. We mix it with saliva. that's conscious. We roll it into a ball. Pay attention as you do this. It's weird. Like once you go over this and then you're like, oh, shit. Roll into a ball, and then we throw it with our tongue to the back of our throat. Right? So we force it to the pharynx. If you notice, we have all of these nerve endings here on the palate and within our oral pharynx that are going to detect food coming in there. What's going to happen when food goes into those areas? Those areas are going to stretch, right? Remember, stretch is one of the stimuli for muscular contraction. And then we go into the swallowing reflex phase. Anytime you hear the word reflex, that's not under conscious control, and it does not require your brain to work it. All right, so the swallowing reflex phase, soft palate, stretching will close off, so it closes off the nasopharynx, and nothing goes up your nose, unless you laugh, right, while you're drinking milk, and the milk comes out, like we've all seen that before, or beer. Where was I? Epiglottis closes, so the epiglottis is in this position. Once it gets a signal, it's going to close and close off the trachea, so food doesn't go down the airway. And then muscles are going to contract because, again, they stretched. Muscles are going to contract because that's what did this. So they're doing this right now. And contracting and pushing the food down into the esophagus. All right, that is a reflex. Stretching, movement up, movement down, closing off passageways, and contracting food down. All right, let's see if there's anything new on here. Yeah, so again, the sensory input from the pharynx, sensory input from the esophagus, something back there, it's stretching. It also inhibits your respiratory center. So while you are, the epiglottis closes off your airway, what can't you do? Breathe. So your diaphragmatic muscles are also going to be inhibited for a short period of time, because what's going to happen if you try to breathe while that's closed? Right? Nothing's going to happen. All right. So we also refer to the swallowing reflex phase. Yes, I did forget the name of it correctly. As the pharyngeal phase. Okay, food in the pharynx. Sensory input from the vagus and glossopharyngeal nerves go and stimulate the muscles to move that food down into the esophagus. So you move from the pharynx into the esophagus. And then we enter the peristaltic phase. Because once you push it into the esophagus stretches, and then we have that receptive relaxation occurring ahead of the bolus of food, with a contraction occurring behind it. It just slowly squeezes down and squeezes down and squeezes down until it gets to the stomach. Is it okay if I erase this?
You know I'll make it look good. All right?
Okay, so we pushed food down. It's to here, but we still have like little scraps hanging to the wall. What happens with those? Right? Especially if you guys ever taken pills without water and like they get stuck back there and you can feel them, you're like, oh my God, go down. What happens is that you continue to have these peristaltic waves that are trying to get it to come down. Right? That's something you don't have voluntary control over. It's going to try to push it down, and they're going to keep going and keep going and keep going until everything gets down. Okay? All right, so that's the esophageal phase. All right, we have two types of peristalsis. One is primary peristalsis, which is very short lasting, and that's a continuation of what happens in the pharynx, so that's that reflex contraction. And then secondary peristalsis takes over, and that's where the distension stimulates muscle contraction and the food goes down by the receptive relaxation and pushing behind. All right? So the upper esophagus, because of the way it works, is striated muscle. What does that mean? It's under somewhat voluntary control, so you can like try to swallow things, right? But the lower is 2/3 of the esophagus is smooth muscle, which means that's going to happen under the autonomic control. All right, I have a video showing you this. It's out on Canvas, but I've already talked about everything, and it talks about this part too. So I'm not going to show it in class, just for time's sake. If you guys want to watch it, just a nice video of the swallowing reflex. All right, so what is the esophagus? In terms of your whole tube, I like to call it the food tube, because this is the only place where actually it's going to be food, right? Because it hasn't entered the stomach, it hasn't been chemically digested, it's just broken down food. All right, so, excuse me, it's muscular, has to go through the diaphragm. You guys know where it goes through the diaphragm, right? That anatomy. And the spot where it goes through the diaphragm is called the esophageal hiatus. So when somebody said, or when you say somebody has a hiatal hernia, that means part of the stomach has come up through the esophageal hiatus and is above the diaphragm. Okay, that would be a hiatal hernia. And what's that going to do to your digestive processes? Do you think? If you have part of your stomach up around here, up here. Pardon?
It's not going to work as well.
Things aren't going to want to go down as well. It's going to compress the esophagus and also make this opening smaller, so things aren't going to want to go down. Plus, it's going to keep that lower esophageal sphincter, LES, lower esophageal sphincter, so that's closed, and when peristalsis comes and knocks up against it and stretches it, it opens. But if you have a hiatal hernia, that's going to remain a little bit open, and what's going to happen to the stomach acid reflux. So you're going to have reflux problems as well. All right, we call the stomach now that we're here, a J-shaped organ. I mean, and I guess it kind of looks like a J. And everybody has a very similar shaped stomach. What did I just read? All right, I don't remember. That's very muscular. In fact, this is just the muscular layer. You can see there are 3 layers of muscles instead of 2, and I mentioned that earlier. You still have the circular that's going to squeeze the stomach like this. You still have longitudinal that's going to shorten this way and propel food down. But then you have oblique fibers that mix in a different direction, because one of the main jobs of the stomach is to do what? Mix food, mix everything together. So you got a lot of juice in there, you got a lot of stuff coming into the juice, you got to mix it all up. Because when it gets to the duodenum, the next part of the digestive tract, you want things to be a homogeneous paste. It has to be a homogeneous paste. If you still have a cookie left over going into the duodenum, it's going to come out, right?
Are the wave fibers, are they superficial, deep in that order, or are they just kind of crisscross?
Say that again.
Like a circular on top of longitudinal on top of oblique, or are they kind of all mixed together?
So remember the circular was, you have the mucosa, submucosa, circular, longitudinal, and oblique. Yeah, so that would be good. Okay, we divide our stomach, so this would be the inside, into 4 different regions. The cardiac region of the stomach, and it's called the cardiac region because it's the closest region to the heart. Like, to me that's a stupid name, but whatever. Then you have the fundus of the stomach. So a fundus is like a bump. So a bump that goes up. And then the rest of it would be the body. So the body of the stomach, and then you have the pyloric region, the 4th region that's just before you enter the duodenum of the small intestine. All right, if you dissect the stomach, and you're not going to see this in the anatomy book, which is one of the unfortunate things about digital dissection. There are a bunch of folds in the stomach. Has anybody dissected a cat though, or in a human and seen the inside of a stomach? Where there's all these folds called rugae. And the purpose of rugae is, 1, to allow for expansion when food enters. Right? If something's folded, then that means it has room to get bigger. And it also acts like a washboard function. So when stuff mixes across it, it like separates it really well. Okay, so anyway, yeah. So kind of like a walk, like a cheese grater, I guess. Like if you move cheese across a flat surface, it's not going to do a whole lot. But then if you got a bunch of bumps to it, it's going to start to crumble. So same thing's going to happen to food stuff within here as well across those rugae. It's really interesting with cat dissections, because this is what we're doing in the undergrad world. You can, outside of what's actually in the stomach, you can tell if the cat has then I'll tell you another story. You can tell if a cat has recently eaten, despite what's in the stomach, if you don't see rugae, because it's all stretched out. And if you look at the stomach, it's going to be very smooth. But if it doesn't have anything in the stomach, then you're going to really see the rugae, which means nothing was in there. Same thing with the urinary bladder. If you open up the urinary bladder and it has a bunch of rugae in it, that means they probably had an empty bladder when they died. But if it's like big and distended, that means they probably had a full bladder when they died. All right. So between the opening and the exit of the stomach, you have the lower esophageal sphincter and the pyloric sphincter. All right. So those are gateways that allow food to enter the stomach but not go back up, and control the exit of food, or what we call at this point chyme. Once food enters the stomach and gets all mixed up, it's called chyme. So controls the amount of chyme that's exiting the stomach. That's what the pyloric sphincter does. So normally when it closed, because the stomach has to do its job, then when it needs to exit the stomach, then the pyloric sphincter is going to control that. So those are 4 regions, 2 sphincters, rugae. We also have, we divide the stomach into 2 regions: the orad region, more towards the oral end, and the caudad region, more towards the tail end. So the opening of the stomach and the tail of the stomach. All right? So like everywhere else, things going in the stomach, receptive relaxation. Most of the mixing occurs down here because, why? Gravity. Right? But then it squishes it back up this way. Makes it go across the washboard, helps things mix up. So that's what we call retropulsion. Oops. All right. So remember early on when we talked about the different layers of the digestive tract, And we talked about the mucosal layer, and it's different in each region. So in the stomach, we said it's geared towards secretion. So what types of things does the stomach secrete? All right. The answer is it depends on where you are. If you're in the ORAD region, what we also refer to as the actinic region, I know, because there's oxalic cells there. That's why it's also called that. We have acid being secreted, pepsinogen being secreted, intrinsic factor, and that's for life. That's probably the most important thing that's secreted by the stomach is intrinsic factor. Does anybody, before we go on, know what intrinsic factor's function is? Ultimately. Nobody remembers? Absorption of B12. Good. So it's important for the absorption of B12. As well as mucus, because you have to protect against the acid and the pepsinogen. Then in the pyloric region, this is where you secrete gastrin, which is a what? Hormone. Okay, so gastrin is secreted by the pyloric region, as well as mucus, because you got to protect, and small amounts of pepsinogen that we're not even going to consider as a secretion for our purposes. All right, so if we look up top, the fundus, the body of the stomach, and the cardiac region, there are three types of cells within what we call gastric pits. All right, so if we look at a micrograph of the stomach, it's going to kind of look like that. And this, I'm writing the textbook here, would be like a gastric pit. So it's this. And so notice that it's lined by a bunch of cells, and these are the cells that are going to be responsible for secreting those substances. All right. In the neck of this gastric pit, we have mucous neck cells, which are going to secrete mucus. That makes sense, right? And that's going to go up and provide a protective function. Down towards the middle, we have the actinic cells, which is why it's called the actinic region, right? Just to kind of put meaningful places. They're going to secrete hydrochloric acid as well as intrinsic factor, so the acid and intrinsic factor. And then down towards the base, we're going to have peptic cells that are going to secrete pepsinogen. Now, pepsinogen, does anybody know what that does? Nope. The answer is absolutely nothing. Because pepsinogen is what we call a zymogen. And zymogens are inactive enzymes. When it goes into the stomach, and comes in contact with hydrochloric acid, it gets turned into pepsin. And pepsin is our main protein in the stomach, from the stomach, that digests proteins. So it's a protease. All right, so that's what's secreted here. Why do we want pepsinogen secreted from here and not pepsin? You'll destroy your own cells. So we secrete it up into the stomach, up into the lumen of the stomach as an inactive precursor. So in the stomach it gets activated, so now you can digest food instead of yourself. Okay, so that's the reason for that.
What was the term for that again?
Zymogen. Zymogen. Once a day zymogen. Okay? It's different when you get down to the pyloric region here. The cells look a little bit different. So this is, I pulled this picture. Here's the link if you want to go read the article. This would be what the oxyntic region looks like, or the ORAD region looks like, and all different cell types secreting the things that we talked about. This is what the pyloric region would look like. So you notice fewer different types of cells, but they're secreting very specific things. One of them is gastrin, right? So they're going to be secreting the hormones. And then also mucus for protection and small amounts of carcinogen. All right, so even different morphology. You're not going to, the gastric pits aren't going to be as deep as what you would see in the rest of the stomach. Just for clarity's sake, I think I've told you guys all of this. You read 5 different textbooks talking about one organ, you're going to see 5 different names for that organ. Okay, so that's what makes some of this really difficult. Corpus would be referring to the body of the stomach, that oxyntic region or the ORAD region, right? 3 different names within one PowerPoint presentation. And the antrum would be this pyloric region. This is called the antral region of the stomach. All right, so what do these secretions do? Why are we secreting this stuff? Well, let's talk about each one individually, then all of them together. So gastric acid has 3 functions. One is it slows down the growth of bacteria. Right? Bacterial, actually no. Yeah, bacteriostatic, and you're going to hopefully in pharmacology or and maybe in your clin med classes, you have bacteriostatics and bactericidal drugs. Bacteriostatics slow down the division of bacteria. Bactericidal kill the bacteria. Okay? So it has a bacteriostatic function. The acid converts pepsinogen to pepsin, right, which we talked about over there, which then begins protein digestion. All right, so where does carbohydrate digestion begin? Mouth. Where does protein digestion begin? Stomach. We talked about this. Here's a picture of what that looks like. So we secrete pepsinogen. Here's how I like to look at it. Although I don't think this is 100% true. Pepsinogen, you cut it, and then you have pepsin. Right? So, like, word puzzles. Maybe it's like 2 word puzzles. We're going to play that Wordle every day. Like, it's like the first thing I do when that's what gets me out of bed. I'm like, all right, but I did my Wordle for the day. Time to get up. So, again, pepsin, proteolytic enzyme, or protease, breaks down protein. Optimal pH is very, very low. What's the normal pH of our stomach when it's empty? 2. pH of 2. So for pepsin to work, you need a lot of hydrogen ions to activate it, so 1.8 to 3.5. If we get our pH up to 5.0, how do we raise the pH of our stomach? How do we typically raise the pH of our stomach? And we do it every day. Eating. Like when we eat, the pH of our stomach raises. In fact, that's one of the signals to start releasing acid, is the pH goes up. But when the pH reaches 5, it can be reversibly inactivated. So if you bring the pH back down, it'll start working again. But if the pH gets up to like 7 or 8, like starts to get basic, you denature the protein and can't get that function back. Right? So there are points in which it can be activated and deactivated. This also is going to be important later on when we talk about acid-base balance. Why do we want acid-bases to be balanced in our body? Because if you get too acidic or you get too basic, what does that do to proteins? Denatures them. All right, so that was acid. Now we're moving on to pepsinogen. What causes the secretion of pepsinogen? 2 signals. 1, vagus nerve, secreting acetylcholine. via the parasympathetic nervous system. And direct responses to gastric acid. All right, so gastric acid can tell those cells directly, say, hey, secrete pepsinogen, I want to do some work here. All right, lots of words. This is an important slide, though. Intrinsic factor. All right, we know that it is released in our stomach by the same cells that secrete what? Is it up there? No. Do you remember? Acid. By the same cells that are secreting HCl. Okay. In gastric juice, the B12 is bound to a protein. All right, and there are a bunch of different names for that protein: haptocorin, R factor. There's even a couple other names. We'll go with R factor. All right. When it gets to the duodenum, what happens to the pH of the chyme that's getting into the duodenum? It increases. It starts getting more basic. All right. When that happens, intrinsic factor is released from R factor. All right. So now it's free. Wait a minute. Back up. No. Okay. B12 is bound to R factor. Did I? I thought I said intrinsic factor is bound to R factor. All right, so B12 is bound to R factor. In the acidic conditions, when you get to basic conditions in the duodenum, R factor releases B12 and intrinsic factor picks it up. All right, so acidic conditions, B12, R factor. Basic conditions, B12, intrinsic factor. And it continues down from the duodenum all the way through the ileum until it gets to the distal ileum where B12 is absorbed. All right, so intrinsic factor carries B12 from the duodenum to the distal ileum. What do we mean by distal ileum? Pardon?
The end.
The end. Okay. They say that this is the only indispensable substance in gastric juice. That means you can live and you can survive and do things without acid, without pepsin, but if you're missing intrinsic factor, you'll die quickly. All right, so you have to have intrinsic factor, which is why people who have stomach reductions need a lot of supplementation and need to be very careful with what they eat and how they treat what's left of their stomach because they need that intrinsic factor. Okay, so if we have ileal disease, preventing absorption, or we have gastric resection like I just talked about, or atrophic gastritis where we don't secrete intrinsic factor, we get an anemia. If we get an anemia, we're sick, right? So that's the importance. Because why do we need vitamin B12? Given what we just said here, what's the purpose? What's the major function of vitamin B12? Making red blood cells, right? B12 is really important for making red blood cells. So we have to have a very highly regulated way of carrying it to where it needs to go. All right, so those are the three things. Mucus is constantly being secreted to provide a barrier, okay? How do we regulate gastric secretion? All right, first of all, what's the purpose of gastric secretion? Why do we need it even? Why do we even need it? Why do we need secretions in our stomach? To break down food in general, right? And turn it into that chyme. So we need to be regulated. So we have to have certain things that regulate it. and they're going to be associated with food getting to our stomach. So first we'll say that somatostatin, which is released from cells in our stomach, it's actually the same somatostatin that's released in your pancreas. Okay? Inhibits acid secretion. So we have to have a way of shutting things off. So we'll start with that. We can shut this process off. We can shut off gastric acid secretion with somatostatin. Acetylcholine, through the vagus nerve, stimulates gastric acid secretion. That should make sense, right? We know that the vagus nerve regulates most of our digestive activities. Also gastrin. Gastrin release. Gastrin, again, is a what? Hormone that goes into the bloodstream, goes back to the heart, comes back, and then affects the cells to secrete acid. Okay, so it gets into the bloodstream. It doesn't go directly to those cells. It gets into the circulation first, then comes back. Histamine also increases gastric acid secretion. So, 3 things: parasympathetic, gastrin, histamine. Okay, even more specifically, from the parasympathetic nervous system, specifically high acid, mucus, gastrin, and pepsinogen. Right, all of those are gastric secretions. Now, we've included gastrin in here, but these are going to be exocrine secretions, right, directly into the stomach, where this is an endocrine secretion. So we're still talking about gastric secretions, but 2 different types. Right? So we'll go from here. Gastrin then can stimulate more hydrochloric acid secretion. In fact, up to 15,000 or 1,500 times more than what histamine can. All right, histamine is specific for hydrochloric acid. So if we want acid, we want histamine and gastrin. If we want the other stuff, we want acetylcholine. We want the parasympathetic nervous system.